Hi everybody, welcome to the third video for Principles in Neural Representation, Lecture 1. In this video I want to just consider some of the limits of this detector analogy that we've been following up till now. When we look at the individual brain cells and consider each individual cell, we've got this idea that the cell might act something like a detector, so the firing rate of the cell seems to indicate something about the presence of a feature in the external world. That idea seems to have some kind of validity, and I was able to give you several different examples from all different kinds of brain systems in the previous video of cells whose firing rates varied systematically according to what was going on in the outside world. So we had the simple visual cells, we had the place cells whose firing rate tells the animal where it is in the environment, we had head direction cells. I also talked about motor neurons and shape neurons. So maybe there is a sense in which we can think about individual brain cells representing information in some way, but I think that does lead us in towards certain misconceptions. And as we go further into the course, I think we're going to need to consider multiple neurons at the same time. So the main purpose of this last video is to connect up to next week's lecture when we'll be thinking about that in more detail. So what are the limitations of this detector analogy? I think there are two issues really. The first issue is that the detector idea might suggest that we kind of need a specialized brain cell for every conceivable percept, concept or action. Anything we might want to do, think, say, feel, every single thing needs a single brain cell ready there to represent it. And is that really plausible? And a second issue is that when we see the firing rate of these neurons, does it really tell us enough to know exactly what we're experiencing? So let's consider the first one of those points. At various times in history, psychologists and neuroscientists and philosophers have kind of entertained the idea that the brain might have a separate neuron to uniquely detect and represent every object or concept. And that idea is called the idea of the grandmother cell. And I know you've thought about it already, and in fact you considered it in quite a lot of detail in teaching block 6 with Daniel Kaiser. There are all kinds of problems with the idea of a grandmother cell, and in fact the concept of grandmother cell was in a way invented to illustrate some of these problems. But one issue is, like, how many brain cells would you actually need in order to represent all of the things that you might encounter in a lifetime, even things that have never existed before? And another problem would be, like, what would happen if you lost the special neuron that you use to represent a particular thing, like your grandmother, for example? There's also a problem of how could you ever test the idea that a grandmother cell exists? Experimentally, how would you find that one cell that represents a specific concept? If you had a recording electrode in the brain, what are the chances of it happening to be right next to the cell that represents Jennifer Aniston or The Simpsons or my granny? So some of the original researchers associated with the Jennifer Aniston cell studies carried out some maths to try and work this out, and by their estimate a given medial temporal lobe neuron would fire to about 0.5% of possible stimuli. But to put this into some kind of perspective, it would still mean several million cells potentially responding to any given stimulus, and each neuron participating in the representation of maybe 50 or to 150 different objects. In other words, the Jennifer Aniston cell phenomenon gives us some evidence of a very sparse and invariant type of representation, but it doesn't really correspond straightforwardly to a single detector for every object or concept we might want to consider. While relatively few neurons are responding to each stimulus, it could still be millions of neurons responding to a given object. And while each neuron responds to relatively few objects or concepts, depending on how you define them, it could still be dozens. So we can't really think about neural representation in terms of individual neurons acting as detectors. We can't really, an individual brain cell can't capture everything that we need to know about in order to understand how the brain is processing a given concept, a given object, a given thought, a given percept, a given action. And the second issue, to come on to that now, is that we haven't really thought about what the firing rate of the neuron really means. Up till now, we've just said, well, it's like a smoke detector. It goes off when a particular conjunction of features is seen in the input. And that's kind of true in a way, but if you think about a smoke detector, it's either not going off or it's going off. It's silent or it's beeping. But neurons don't work like that. Although each individual action potential is a kind of all or nothing event, what we've seen is that the neuron signals the presence or absence of an external feature in terms of its firing rate, and it has a tuning curve. So how do we interpret variations in the firing rate in terms of what the cell is signaling? Is that useful information, and what ambiguities does it leave over? So here are three examples of the cells that we considered earlier on in the previous video. On the left you've got a V1 simple cell that indicates the orientation of an edge in the visual field. In the middle we've got a head direction cell from the postsubiculum of the rat that indicates which direction the animal is heading. And on the right you've got a motor cortical neuron from the motor cortex of a monkey, where the firing rate indicates the direction that the monkey's hand is going to move. Now, we've really mainly just thought about the preferred value of the cells up till now. So we've been concerned with what makes the cell fire maximally. So for the V1 cell that you can see illustrated there, it fires most strongly to a vertical edge. But what about when it's firing less strongly? What if its firing rate is to one of these oblique edges? What you can see is that in this diagram, at least, two very different looking edges can produce the same firing rate. So if we actually saw that V1 simple cell firing and we knew nothing else, we would not know whether the edge that we were looking at was orientated like this or like this, which is quite a lot of difference. There is different orientations as you can have of a bar. Same with the head direction cell in the middle. Let's imagine that we see that the head direction cell is firing and it's firing at about 40 hertz. Okay, which direction is the rat heading in? It could be heading at, let's say, 200 degrees, or it might be that the firing rate is indicating that it's heading at 320 degrees. So completely different directions in the environment. So the motor cortical neuron might be even more telling, and you're going to get a chance to think this through for yourself in the tutorial that's coming up, and we will talk about it a little more next week. But these kind of neurons are supposed to control the direction that the animal is going to move its hand. So in this case, the neuron has a preferred direction of 180 degrees, so that corresponds to moving its hand to the left. Okay, now imagine that the cell isn't firing at its maximum firing rate, but is firing at half its maximum firing rate as indicated by those red arrows there. Well, what would happen then? We can't really tell whether the hand is going to move in, say, 45 degrees, which would be like this, or is it going to move at an angle of 270 degrees, which would be like this. So two very different actions corresponding to the same firing rate. So an individual brain cell can't really tell us which way the animal's hand is going to move. So the point of this part of the course is principles of neural representation. And the principle that seems to be at work here is that there are some ambiguities in the output of any individual neuron that has a bell-shaped tuning curve. We've seen these bell-shaped tuning curves in quite a few neurons across different parts of the brain. And what they tell us is that we can't always interpret the firing of an individual neuron very, very accurately and precisely. And that's what's illustrated in this diagram. What we've got here is the classic bell-shaped tuning curve. So on the x-axis, we've got some parameter, some external feature of the world that's being represented by this neuron. Now, I guess you could think about the orientation of a bar in the visual field, or you could think about the location within a room, in a place cell, for example, or you might even think a little bit more abstract, oh, is this the identity of Jennifer Aniston? But wherever we have a neuron who's got a kind of bell-shaped tuning curve like this, there's some ambiguity in what it's signaling. So you can see these red areas to the left and right of the diagram. Those indicate regions where the cell isn't firing at all. So presumably there's going to be a great deal of uncertainty about what's happening in that space. The neuron isn't telling us very much about where we are in that space. If the cell isn't firing, we don't know where we are. Similarly, if we look at the white region in the middle of the plot, there are always different parts of that curve that can be confused with one another if we're just thinking about one neuron. So if we look in the middle of the plot, quite near to the preferred value, points B and point C, they have similar firing rates, so we can't really tell them apart from the firing of this individual brain cell. Similarly, if we look at points A and points D, they're quite far apart. We really can't tell them apart. The firing rate of the neuron would be expected to be the same in both of those very different cases. And that might mean confusing an orientated bar like this with an orientated bar like this, or it might mean confusing my grandmother with your grandmother. And perhaps because of the way that we've been thinking about individual neurons as detectors up till now, we've tended to see this broad tuning as potentially problematic. It introduces that kind of ambiguity. Okay, well, we don't know exactly what we're looking at. We've got some uncertainty about what's being signaled by an individual neuron. But maybe that kind of information could also be useful in some ways, because it means that the cell can communicate something useful about the outside world, even when the conditions that it's tuned to detect aren't being precisely met. So it means that the cell can still send out a signal even if the line isn't quite orientated in the right direction. It means that we can still recognize Jennifer Aniston even though she's wearing a different dress on this occasion or is being lit in a different way. So the broad tunings of these cells might mean that it's possible to generalize across different examples, different types of stimuli. It means that we perhaps don't need a neuron for every single stimulus that we can encounter. So the breadth is important. And the fact that a neuron's firing rate varies continuously rather than in an all or nothing way means that the firing rate itself can carry some useful information about the external world. So it can signal the degree to which a stimulus matches its preferred value, or the probability to which a stimulus is present, or the degree to which something appears to be true, or the extent to which a particular action is planned or intended. And you can kind of get a feeling for this by looking at the picture on the right hand side here. If you've got a neuron in your brain that's tuned to detect rabbits, then that cell probably should be firing a little bit when you look at that image. But also maybe the duck detector in your brain also needs to fire at the same time, and maybe there's some degree of uncertainty about what you're actually seeing there. So that uncertainty could potentially be reflected in the firing rate of the different neurons that are involved in that representation. So in principle, the firing rates of neurons can represent and reflect uncertainty. So today's lecture has been about representation in individual neurons. Neurons at this level can be thought of as acting a little bit like detectors, and in that sense they represent the thing that they detect, and they signal that detection by varying their firing rates. And you've seen examples from across the brain, from sensory, motor, but also from cognitive representations, and we've seen the same pattern in several cases: bell-shaped tuning curves, broad tunings to one or more stimulus dimensions. We've also seen examples of very sparse and invariant representation in individual neurons that respond to a single concept, like the idea of Jennifer Aniston. But even these cells are likely to fire to multiple stimuli. And the broad and graded responses that we see in individual neurons, rather than just merely detecting something, allow for some generalization and give a useful indication of how well a cell's preferred tuning characteristics are being matched. But that can lead in turn to some ambiguity in the interpretation of an individual brain cell's responses. So next week we're going to look at how information from multiple neurons can be combined and how it can capture a much richer picture of the world around us and resolve that ambiguity in the output of individual neurons.